continuity refers to reproduction, the passing on of genes from one generation to the next. For cells, this means making more or new cells, and based on cell theory, all new cells come from pre-existing cells, and that requires cell division. Here's a quick question. Pause the video and think about the answer. If you chose B, you're certainly correct, and if you didn't, that's totally fine. You may want to listen especially carefully to this lecture. Cell division, then, is a mechanism by which one cell gives rise to two new cells. This is true whether we're talking about bacteria, archaea, or eukaryotes. We'll start by talking briefly about cell division in bacteria, and then we'll focus most of our discussion on eukaryotic cell division. Bacteria reproduce by a mechanism known as binary fission. This is described very generally in your textbook, and we'll be keeping it at a very general level as well. You can think about this process as having three major stages. The first, of course, is the replication of the chromosomes. And the mechanism for chromosome replication is really exactly what we've just gone through in a previous module, looking at DNA replication or DNA synthesis. The bacteria will duplicate their genome, and then when they divide, each new daughter cell will get the identical genome to the parent cell. After chromosome replication, the two new chromosomes need to be separated from each other and be placed at different ends of the cell, so that when this cell divides into two, it will actually separate the two genomes. This process has in the past been very poorly understood, but a fair bit of research has been done more recently on it. It certainly involves cytoskeletal proteins, but again, we are not going to go into those details today. Once the two chromosomes have been separated, then the cell has to divide in two. Because it's DNA replication that gives rise to the two genomes that will be separated, these two genomes are genetically identical to each other, and the two daughter cells are also genetically identical. This type of replication or reproduction is known as asexual reproduction, where the two daughter cells are identical to each other and to the original parent cell. This type of division produces clones. I've already mentioned that the chromosome separation stage requires the activity of special cytoskeletal proteins, and the cell itself will also divide with the help of cytoskeletal proteins. One of the interesting aspects of binary fission in bacteria is that each time a bacterium replicates, it actually doubles the number of cells. When one cell divides, it forms two. When the two cells that result divide, that forms four. 4 will form 8, 16, etc. So you're doubling the population size every time these cells go through a round of division. This is known as exponential growth, and explain how bacterial populations can grow so rapidly. Many bacteria can reproduce on the order of every 20 minutes or so you can get really rapid population growth in these organisms. If we now include eukaryotic cell division, we end up with three different types of cell division. In the first case, you have prokaryotic binary fission that we've already discussed. It gives rise to two identical daughter cells. Eukaryotic mitosis is similar to prokaryotic binary fission in that it also produces two genetically identical daughter cells from the original parent cell. So both of these types of division are asexual reproduction. In both cases, once the original parent cell divides, it ceases to exist. The third type of cell division is known as meiosis, and it is found in eukaryotic cells only. Meiosis is different from mitosis for a number of reasons. We'll discuss those reasons in detail when we talk about meiosis, but in general, this involves two rounds of division and not one. The result is 4 daughter cells rather than 2 daughter cells, and these 4 daughter cells are genetically different from each other and genetically distinct from the parent. The genetic information that each one of these daughter cells has is a subset of the original genetic information that was in the parent cell. Meiosis is required for sexual reproduction in eukaryotic cells. Let's now discuss mitosis and put it in the context of what is known as the cell cycle. Mitosis, as we've already mentioned, gives rise to identical daughter cells, or clones. This type of division is used for reproduction among single-celled organisms. Here's an amoeba which is just finishing up cell division to generate 2 daughter cells. In multi-celled organisms, mitosis is required for growth and development. So here we have a sand dollar egg which has been fertilized and has completed the first division, or the first cleavage. These 2 cells will now go on to undergo division through mitosis multiple times in order to ultimately generate an embryo and finally an adult. And then finally, mitosis is also involved in tissue repair and renewal. So some of our own cells, such as our epithelial cells, the various types of stem cells, such as the stem cells that give rise to our blood cells, are undergoing constant rounds of mitosis. If you damage or cut your skin, then this will stimulate higher levels of mitosis in the area in order to repair the damaged region. And if you think about the lifetime of a cell, it does not spend all of its time in division. Early experiments, some of which are described in your textbook, showed that the cell in its lifetime will go through 4 major stages. These 4 major stages form part of what is known as the cell cycle. And the cell cycle simply can be divided into the time it spends dividing during mitosis, or the M phase shown here, and the time it's not dividing, and that is known as interphase, and that is represented by the rest of this circle shown here. But interphase itself can be further divided into 3 separate stages, and again, some of the early data that provided evidence for these 3 stages is described in your textbook. Now, before we describe all of these stages, let's pause the video and see if you can answer this question. This is a pie diagram, and so the size of the pie that's taken up by any given stage represents the amount of time that the cell is spending in that stage. And you can tell by these types of diagrams that mitosis normally takes up a very small amount of the life cycle. The bulk of the cell's life will be spent in interphase. But as I've mentioned, interphase is not always the same. There are actually 3 different stages associated with interphase. This second stage here, with the dark red, is known as the S phase, and this is when DNA is synthesizing. A common misconception is that DNA is replicated during mitosis, but that is certainly not true. It happens prior to mitosis, during S phase. So let's walk through the different stages of interphase. In the G1 phase, the cell is essentially making a living doing whatever it does as a cell. So it will be growing, producing proteins, you'll have transcription, translation taking place, of course, be building lipids, getting larger. And during this time, especially during the last portion of the G1 phase, it is also starting to prepare for DNA synthesis. That may involve turning off some genes and turning on some genes. S phase, or synthesis phase, is the time when the cell is actually actively replicating its DNA, and we talked about that a few modules ago. In addition to DNA synthesis, other activities are happening in the cell, and we'll talk about that in just a minute. Following S phase, when the DNA synthesis is complete, the cell does not immediately go into mitosis, but normally there is another gap phase known as the G2 phase, where it's actually undergoing additional growth. It's taking in nutrients, it's building more organic molecules, it's producing RNA, protein, etc. And during this phase, it's now preparing for mitosis. It'll be making enzymes and other types of proteins that are going to be critical to moving through the stages of mitosis. When everything is prepared and the cell is ready to move into mitosis, it will shift out of G2 and into what is known as M phase. I'll also mention that there is a 4th phase of interphase, which is known as G0. And this is what happens to cells that are not going to be undergoing rounds and rounds of cell division. So we have a lot of cells in our body that do not divide regularly or sometimes at all. And they actually move out of G1 and into a special phase of interphase known as G0. And it's noted as G0 because it means it has in some ways moved out of the cell cycle and has gone into a period where they just continue to exist, metabolizing as a regular cell, doing whatever the cell does for a living, but not preparing for DNA synthesis and ultimately for mitosis anymore. Another point I'd like to mention before I move on here while we're talking about cell cycle is that this entire cell cycle is under strict molecular control. So whether a cell is in G1, is ready to move into S phase, is going to go from S to G2, and whether it enters mitosis at all will be dependent on molecular signals that exist inside the cell. It involves the turning on and off, or the activity of different genes as well as different proteins. This is especially true in multicelled organisms, where tissues and organ size have to be strictly controlled. We're not going to discuss the molecular control of the cell cycle in this course, but your textbook does provide an introduction to this topic if it's something that interests you. Not surprisingly, the control of cell division in multicelled organisms involves various signaling pathways, and problems with those pathways or mutations of proteins that are critical in these pathways can lead to various types of disease, including cancer. Let's go back and look a little bit more carefully at S phase or synthesis. Certainly S phase is a moment when DNA is synthesizing, so DNA replication is happening. Histones are being translated very quickly because large numbers are needed in the nucleus to complex with the new copy of the genome. But other things are also happening in preparation for mitosis or cell division. You'll remember that in animal cells, they have two centrioles which are found at the heart of the microtubule organizing centers. And these microtubule organizing centers are usually found near the nuclear envelope. During synthesis, or S phase, the centrioles also replicate. It's not just the DNA, but we're also going to need two poles inside the mitotic cell, and that means we need two sets of centrioles. This also will ensure that when the cell divides, each new cell will receive two normal centrioles, so that each new cell will also have a functioning microtubule organizing center. The third thing that's happening in terms of synthesis during S phase, as I've already mentioned, will be the translation and the synthesis of proteins which are important to complex with DNA in order to form normal chromatin, which of course will include the histone proteins. Before we talk about mitosis, I just want to point out a little bit about terminology and a few extra structures that we need to be aware of. First of all, when we talk about the eukaryotic chromosomes, you'll remember they are linear and they have telomeres at either end. When we're talking about mitosis, we often show a single chromosome as a line shown here. This often results in students thinking about the chromosome as a single strand of DNA. But in fact, this line, when we're talking about chromosomes, really denotes a double-stranded helical DNA molecule. And then somewhere along the eukaryotic chromosome, there will be a special region known as the centromere. This centromere is identified differently in different organisms. In some organisms, such as yeast, it's a specific sequence of DNA. This centromere will be a critical structure for mitosis because it is at that structure that the kinetochore is going to form, and that's the part of the chromosome which is going to connect directly to the spindle. In S phase then, DNA replication results in two DNA double helices. And again, we often show them as two lines, but remember they're not single strands of DNA, they're each a double-stranded helix, as shown in this image here. Because these two replicated chromosomes have been generated through DNA replication, they should be genetically identical to each other, and we call those two DNA molecules sister chromatids. As the cell transitions into mitosis, you'll see that DNA become increasingly folded up and condensed to form what we're accustomed to seeing as chromosomes. These two sister chromatids are held together tightly at the centromere region, and this is where the chromosomes appear to be pinched in. Right there. This is the centromere region right here. So one sister chromatid along the top and a second sister chromatid along the bottom. Now to bring in a little bit of terminology. Prior to S phase, of course, we have each chromosome consisting of a single double helix of DNA. If this was a human cell, then of course it would be 46 chromosomes in the nucleus. After S phase, we've got two sister chromatids for each chromosome. We've doubled the amount of DNA. Each of these sister chromatids, however, will be held together. And as long as they're held together, they're considered to be a single chromosome. As a result, the chromosome number does not change following S phase. So the number of chromosomes in the cell is the same in G1 as in G2, even though the amount of DNA has doubled. When these sister chromatids are separated, which is going to happen in anaphase of mitosis, they will become individual chromosomes in their own right, and this allows each daughter cell to receive a full complement of chromosomes. Well, I've mentioned that the sister chromatids are held together until anaphase. How are they held together? They're held together through the activity of special proteins known as cohesins. These cohesins seem to be able to form rings around the two molecules of DNA. These are two double helices of DNA, and they hold the sister chromatids together throughout the rest of S phase into G2 and into mitosis. The cohesins will not be broken down until anaphase or just before anaphase to allow the separation of the sister chromatids in anaphase. And just to reiterate, because this is just a critical concept for you to understand the terminology, etc., if you've had difficulty with mitosis and meiosis in the past, this is your moment to clear it up. If you've never had difficulty, then you can skip through this part. Alright, so we start off in G1 prior to S phase as single DNA molecules. Each chromosome consists of one molecule of DNA. S phase includes DNA replication, so each chromosome at the end of S phase consists of two identical molecules of DNA, two identical double helices of DNA. These two sister chromatids are held together by special proteins known as cohesins. By definition, these two sister chromatids being held together by cohesin are considered still to be a single chromosome. During anaphase of mitosis, these two sister chromatids will be separated, with one going to each new daughter cell. As soon as they separate, each of these sister chromatids are considered to be a chromosome. Cell division in eukaryotes has two major stages. The first is known as karyokinesis, and this really refers to the division of the chromosomes or the nucleus. So we're dividing up the nuclear material, and this by definition is mitosis. But in order to get two daughter cells, you also actually have to divide the physical cell into two new cells. This process is known as cytokinesis, or the division of the cytoplasm. In the next lecture, we'll talk in a little bit more detail about the two stages, mitosis and cytokinesis.